Hello, everyone. Good day. For this lesson, the focus of our discussion would be on the family Neisseria and related organisms. To start with, let's discuss the general characteristics that are commonly shared by members of the family Neisseria. Take note that generally, members of the family Neisseria are aerobic, which means that they require oxygen for growth. They are non-motile simply because they lack the organelle that enables bacteria to move, which is the flagellum or flagella, and they are non-spore forming bacteria, which means that they cannot form endospores in response to harsh environmental conditions because, as we all know, there are only two pathogenic genera that are capable of doing such, which are Clostridia and Bacillus. And typically, members of the family Neisseria, particularly the genus Neisseria, are gram-negative diplococci. As you can see in this microscopic view of a specimen that is subjected to direct microscopy, you can see here gram-negative diplococci. Gram-negative in the sense that they are able to retain the safranin counterstain, and they are cocci because of their spherical appearance. However, they are diplococci because they are arranged in pairs, and take note that its distinguishing characteristic includes the presence of adjacent ends that are flattened. As you can see in this electron micrograph image, the adjacent ends of each coccus that make up the diplococci are flattened, giving the organism a resemblance to tiny coffee beans or kidney beans. So in this microscopic appearance, you can observe the presence of gram-negative diplococci that have ends or adjacent ends that are flattened. However, an exception to these characteristics include Neisseria weaveri, Neisseria elongata, and Neisseria bacilliformis, which are rod-shaped members of the family Neisseriaceae. Also, generally, members of the family Neisseriaceae are catalase positive, which means that they possess the enzyme catalase that converts hydrogen peroxide into water and oxygen, resulting to effervescence or bubble formation. And they possess cytochrome oxidase. However, an exception to this are the following: Neisseria elongata subspecies nitroreducens and Neisseria bacilliformis. In terms of growth requirements, take note that members of the family Neisseriaceae are capnophilic. As learned previously, capnophilic bacteria require increased concentration of carbon dioxide, around 3 to 10%. And on top of this, members of the family Neisseriaceae are humidophiles, which means that they require increased moisture. In the laboratory, how do we achieve a capnophilic environment that is supportive for the growth of Neisseria? Routinely, we can use a candle jar. So in this setup, after culture plates or culture tubes are placed in the jar, a candle is lit. And once the cover of the jar is placed, it displaces oxygen and it would extinguish the flame. And once the flame is extinguished, smoke forms that supplies the required carbon dioxide concentration needed for capnophilic Neisseria to grow. And take note that in this setup, moisture also forms in the jar, and that moisture is enough to provide sufficient humidity that is needed for the growth of Neisseria. Commercial systems are also available that can be used to support the capnophilic environmental requirement for the growth of Neisseria. We have CO2 biobag type C and CO2 Gen Compact. And there is also what we call a carbon dioxide incubator that is capable of maintaining a 3 to 10% CO2. And we can also put a pan with water because during incubation, moisture forms in the presence of the water in the pan that is enough to support a humid environment for the growth of Neisseria species. Take note also that the optimal temperature requirement for the growth of pathogenic Neisseria is between 35 degrees and 37 degrees Celsius, although some books would say it's between 32 degrees to 36 degrees Celsius. The natural habitat of Neisseria species are the mucous membranes of the respiratory tract and urogenital tracts. So take note that even among healthy individuals, Neisseria organisms inhabit in these areas as commensals. However, take note that with the wide range of species under the genus Neisseria, there are only two organisms that are considered as primary pathogens. We have Neisseria gonorrhoeae and Neisseria meningitidis. So these two organisms will be the focus of our discussion as we move along with Neisseria species. Take note that Neisseria gonorrhoeae, also known as Gonococcal, and Neisseria meningitidis, also known as meningococci, are highly fastidious. What does this mean? This means that these primary pathogens under the genus Neisseria require sophisticated and additional nutrient requirements for them to grow. Take note that bacteria require nutrients for growth. However, what sets apart Neisseria gonorrhoeae and Neisseria meningitidis from other bacteria is that they require additional nutrient requirements that must be supplemented to a culture media to support their growth as fastidious organisms. Specifically, Neisseria gonorrhoeae and Neisseria meningitidis require iron for growth. And because of their fastidious nature, these two organisms, if we wanted to isolate them from clinical specimens, they require the use of enriched media for growth. As learned in the topic culture media, when we say enriched media, these are basal media that are added with specific nutrients to support the growth of fastidious bacteria. In the laboratory, there are two types of enriched media that are routinely used for the isolation of fastidious bacteria, namely chocolate agar and blood agar. Why are these media considered enriched? Simply because they are added with blood and other nutrients to support the growth of fastidious bacteria. However, take note that between Neisseria gonorrhoeae and Neisseria meningitidis, Neisseria gonorrhoeae grows on an enriched chocolate agar plate. When we say enriched chocolate agar plate, these are basically chocolate agar that are further enriched with nutrients to provide the nutritional requirements that Neisseria gonorrhoeae for growth. In particular, take note that Neisseria gonorrhoeae requires cysteine for growth. And a chocolate agar plate could be further enriched with cysteine to make it more nutritive for Neisseria gonorrhoeae to grow. And on top of that, an enriched chocolate agar plate also contains X factor and V factor, also known as nicotinamide adenine dinucleotide or coenzyme 1, which are also needed for Neisseria gonorrhoeae to grow because it is a fastidious bacteria. Chocolate agar plate contains lysed RBCs because, as we learned in our laboratory classes, the RBCs added in a blood agar base is lysed upon heating the medium with temperatures above 70 degrees to purposely liberate X factor or hemine from RBCs, because hemine or X factors from RBCs are important nutrient requirements to support the growth of fastidious bacteria. V factor can also be derived from lysed RBCs. However, some bacteria abundantly produce V factor. And also, V factor can be obtained from yeast, yeast extracts, and potato extracts, and therefore, we can supplement further chocolate agar plates with yeast, yeast extracts, and potato extracts to supply more V factors needed for Neisseria gonorrhoeae to grow. Take note that Neisseria meningitidis only requires shaped blood agar as the minimal growth standard. What does this mean? Between Neisseria gonorrhoeae and Neisseria meningitidis, it is only Neisseria gonorrhoeae that requires X factor. And therefore, since Neisseria meningitidis does not require X factor for growth, shaped blood agar is sufficient to primarily isolate this Neisseria species from clinical specimens. However, I did not say that we cannot inoculate specimens suspected of Neisseria meningitidis using an enriched chocolate agar plate. What I said is that as a minimal growth standard, we can use sheep blood agar alone. So what are those instances wherein we can utilize enriched chocolate agar plate for the isolation of Neisseria meningitidis? Take note that enriched chocolate agar plates that are used for the primary isolation of Neisseria gonorrhoeae contain antibiotic inhibitors that inhibit commensal bacteria and contaminants from clinical specimens to promote the growth of Neisseria species. And clinical specimens that are submitted for the isolation of Neisseria meningitidis can be inoculated on an enriched chocolate agar plate that had been supplemented with antibiotics if those specimens were obtained from anatomical sites that are rich with microbiota. And the only purpose why we inoculate those specimens on an enriched chocolate agar plate is to suppress the normal microbiota and to promote the growth of Neisseria meningitidis. But as I emphasized earlier, if specimens are not contaminated with normal microbiota, and if that specimen is submitted for the isolation of Neisseria meningitidis, then sheep blood agar plates would already suffice as the minimal growth standard. So remember, enriched cap is for the growth of Neisseria gonorrhoeae, while sheep blood agar plate is the minimal growth standard for Neisseria meningitidis. However, in reverse, Neisseria gonorrhoeae grows poorly on blood agar plate because it lacks the necessary nutrient requirements for it to grow, and therefore sheep blood agar plates cannot be used as the primary isolation media for Neisseria gonorrhoeae from clinical specimens. Take note that both organisms, Neisseria gonorrhoeae and Neisseria meningitidis, are sensitive to temperature changes and must be protected from cold. And this is for this reason that specimens suspected of having Neisseria gonorrhoeae or Neisseria meningitidis must be inoculated immediately on this primary isolation media, specifically enriched chocolate agar plate for Neisseria gonorrhoeae and blood agar plate for Neisseria meningitidis, so that they will be incubated using its required incubation requirement, which, as I said earlier, 35 degrees Celsius with an increased CO2 and increased humidity, because Neisseria gonorrhoeae and Neisseria meningitidis, apart from they are sensitive to temperature changes, they are also sensitive to drying, and that explains their need for increased humidity upon incubation. At this point, let's talk about the virulence factors of Neisseria species, particularly the pathogenic ones, Neisseria gonorrhoeae and Neisseria meningitidis. As a review, virulence factors refer to components present on the surface of bacterial cells or components that are secreted by bacterial cells that contribute to their ability to cause disease and contributes to the degree of their pathogenicity. Take note that Neisseria species, particularly the pathogenic ones, as mentioned earlier, requires iron for growth. And therefore, in order for them to survive inside human hosts, they must be able to compete human hosts for iron, and they can do that because they have receptors for human transferrin. Neisseria meningitidis is unique because it is the only pathogenic Neisseria that possesses a polysaccharide capsule. If you're going to look at this illustration here, the cell wall of Neisseria meningitidis has an extra layer of polysaccharide, which makes up its polysaccharide capsule. And as a virulence factor, it enables the Neisseria meningitidis to resist phagocytosis, and therefore they can evade the killing effects of phagocytes. For both organisms, Neisseria gonorrhoeae and Neisseria meningitidis, they have pilus or pili, and primarily the pili of these organisms are used by them to attach to host mucous membranes. So basically, they're able to colonize mucous membranes of the gastrointestinal tract. I mean, they are able to colonize mucous membranes of the urogenital tract or respiratory tract because of the pili that they possess on their surface. However, apart from functioning as attachment organelles that they use to attach to mucous membranes, the pili of Neisseria gonorrhoeae and Neisseria meningitidis also play a role in the conjugation and transfer of genetic materials, such as genes that code for antimicrobial resistance. Take note that Neisseria gonorrhoeae in particular has been known to resist many types of antibiotics that were previously used to treat gonorrhea infections. And their resistance to antibiotics have been acquired via genetic transfer mediated by the bacterium's pilus. Also, the organisms, both Neisseria gonorrhoeae and Neisseria meningitidis, also possess cell membrane proteins that, apart from performing their normal function to the organism, these cell membrane proteins also contribute to their pathogenicity. We have here protein 1, protein 2, and protein 3. Protein 1 is the porine protein present on the outer membrane of Neisseria gonorrhoeae and Neisseria meningitidis. And its normal function is to serve as a channel for passage of nutrients and waste products produced by these two organisms as they metabolize nutrients. And this protein 1 is coded by POR-A and POR-B genes. For Neisseria gonorrhoeae, the protein 1 that is coded by POR-B gene has protective effects for the organism against hosts' inflammatory response and complement-mediated killing, making them capable of resisting the harmful effects of complement as well as the harmful effects of phagocytosis mediated by C3b as an opsonin. Protein 2, expressed on the surface by both of these pathogenic Neisseria, also facilitates their adherence to phagocytic cells and epithelial cells. And protein 3 also enables these two organisms to block host IgG. As discussed in immunology, IgG antibodies play a vital role in opsonized phagocytosis, and the ability of the protein 3 of these two pathogenic Neisseria to block host IgG make them also able to resist phagocytosis mediated by IgG opsonins. Another important virulence factor produced by these two pathogenic Neisseria is the IgA protease that cleaves IgA antibodies in mucous membranes. In immunology, we learned that dimeric IgA populate the mucous membranes and they serve as primary defenses for human hosts against bacteria that enter via the mucosal surfaces. And the fact that Neisseria gonorrhoeae and Neisseria meningitidis secrete IgA proteases that can cleave IgA antibodies that are present on mucosal surfaces, then these antibodies would be destroyed, and as a result, Neisseria gonorrhoeae and Neisseria meningitidis would be able to colonize and establish infections in mucosal surfaces. Perhaps one of the most important virulence factors produced by pathogenic Neisseria is their LOS, or lipo-oligosaccharide, which is similar to the lipopolysaccharides of other gram-negative bacteria, simply because Neisseria gonorrhoeae and Neisseria meningitidis are gram-negative, but their LPS is called LOS, or the structure that they possess is called LOS instead of LPS. But similarly, the LOS of Neisseria species contained the lipid A portion, which is the location of the endotoxin. So take note that the LOS of Neisseria gonorrhoeae and Neisseria meningitidis are anchored in the outer membrane, and the lipid layer, or the lipid A layer of the LOS, is found beneath the outer membrane. So normally, when bacterial cells lyse, particularly when gram-negative bacteria undergo lysis, the lipid A becomes liberated. And since the lipid A portion contains the endotoxin portion, then the endotoxin portion becomes present in the circulation. And in large amount, it would result to endotoxemia. However, even if Neisseria gonorrhoeae are not lysed, but according to the book, during rapid periods of growth, or if there is rapid growth of Neisseria gonorrhoeae and Neisseria meningitidis inside hosts' bodies, their growth releases outer membrane fragments called blebs, and these outer membrane fragments or blebs contain the LOS. And as learned earlier, the LOS contains the lipid A portion, which is the location of the endotoxin. So take note that even if there is no lysis of Neisseria gonorrhoeae or Neisseria meningitidis, the blebs released during rapid growth contain LOS. So when endotoxins that are present in the lipid A of Neisseria gonorrhoeae and Neisseria meningitidis are released, either it could be through lysis of the cells or through the formation of blebs, the large amounts of endotoxins released by these organisms can stimulate macrophages to produce increased amounts of pro-inflammatory cytokines. So during endotoxemia, when we say endotoxemia, this refers to the increased presence of endotoxins in the blood. And as emphasized earlier, it could be released through lysis of gram-negative bacterial cells, or in the case of Neisseria gonorrhoeae and Neisseria meningitidis, the formation of outer membrane blebs during rapid growth result also in the presence of endotoxins in the circulation. And endotoxemia, which is referred to as the presence of high amounts of endotoxins, could stimulate macrophages to produce increased amount of pro-inflammatory cytokines. To mention a few, we have here interleukin-6, interleukin-1, tumor necrosis factor alpha, and other mediators. So take note that normally in the presence of inflammation, These pro-inflammatory cytokines are beneficial as they promote inflammation. And as discussed in immunology, inflammation is actually beneficial as it helps in the activation of the immune response, and it helps localize and eliminate the cause of tissue injury or the cause of infection. However, when there is excessive amounts of pro-inflammatory cytokines produced by macrophages as a result of endotoxin stimulation by macrophages, then the excessive amounts of inflammatory mediators could cause a heightened inflammation. And when inflammation becomes heightened, or basically when inflammation becomes out of control, the effects would not only be beneficial anymore, but becomes harmful to the host. So what are the effects of endotoxins? So as I said earlier, it could result to prolonged inflammation and fever as macrophages have been stimulated by endotoxins to produce large amounts of pro-inflammatory cytokines. And the presence of large amounts of pro-inflammatory cytokines would result to prolonged vascular permeability or prolonged vasodilations. As you can see here, there is vascular permeability or vasodilation. And when there is vasodilation, there would be capillary leakage, meaning to say components of the blood vessel like plasma and blood cells would leak out from the blood vessel to the tissue. And during heightened immune response, this continued capillary leakage would result to purpura and petechial rash. So this is a picture that shows purpura and petechial rash, and this results from prolonged capillary leakage as a result of prolonged vasodilation. And take note that since there is prolonged hemorrhage or capillary leakage, there would be hypotension. When we say hypotension, the blood volume in the circulation would be decreased. And because there is decrease in blood volume, tachycardia would occur, meaning to say that the heartbeat is increased more than 100 beats per minute because it compensates the decreased blood volume in the circulation. Tachycardia increases, or when there is tachycardia, the heart tries to compensate the hypotension by increasing heartbeat so that there would be increased circulation of oxygen carrying RBCs that deliver oxygen to vital organs in the body. But over time, as prolonged inflammation continued to occur because of endotoxin stimulation, there would not be enough oxygen that could be delivered to vital organs. And when there is not enough oxygen delivery to vital organs, it would result for organs to abruptly cease their function. And that is what we call septic shock. And because there is prolonged vascular permeability, then there would also be intravascular coagulation. So these are the effects of endotoxin stimulation by macrophages. So take note that inflammation is a double-edged sword. As emphasized earlier, it is beneficial when it is able to achieve its primary purpose in the immune response. And take note that inflammation's primary purpose include localization of infectious agent and activation of the immune response to help the host fight infection. But when inflammation goes south, such as due to endotoxin stimulation, the heightened inflammatory response as a result from the increased amounts of pro-inflammatory cytokines secreted by endotoxin stimulated macrophages could be damaging to the host. And these are the things that would happen to the host if there is a heightened inflammatory response. At this point, we are going to talk about the disease association of pathogenic Neisseria species. To start with, let's talk about Neisseria gonorrhoeae, which is the causative agent of gonorrhea. So as we all know, gonorrhea is a sexually transmitted infection. So as seen in these pictures, gonorrhea is characterized by an acute pyogenic infection. Pyogenic in the sense that there is presence of draining pus in the infection site, particularly in the urethra of men and endocervix of women. Neisseria gonorrhoeae infects ciliated or infects non-ciliated columnar and transitional epithelium. I'll repeat, Neisseria gonorrhoeae infects non-ciliated columnar and transitional epithelium, which are found in the urogenital tracts of males and females. And when they infect these types of cells, it would result to the formation of pus. Because draining pus is a characteristic manifestation of gonorrhea, the disease has been known previously as the flow of seed because the urethral discharge among infected men was mistaken for semen. Gonorrhea is also previously known as syphilis, or it was previously confused rather with syphilis, because apart from the fact that syphilis and gonorrhea occur in the same individual at the same time, the symptoms of syphilis could also be similar with gonorrhea. In the past also, gonorrhea infection has been known as the clap. There had been many theories as to why the disease was known as the clap. The first theory states that the reason why the disease was previously called as the clap because it was derived from the French word clapier, which means brothel, or in modern time, this is a place wherein men will be able to have access to prostitutes. Other theories also suggest that the clap, which refers to Neisseria gonorrhoeae infection, is attributed to the throbbing pain caused by the infection, or it may refer to the early treatment of gonorrhea that involves smashing or clapping the penis with both hands at the same time to get rid of the pus or the discharge. As I said earlier, Neisseria gonorrhoeae is primarily acquired by unprotected sexual contact and occur primarily in the urethra of men and endocervix of women. However, take note that it may also occur in the anal canal and pharynx for those individuals who practice unsafe oral anal sex. And it may also occur in the conjunctiva. However, the victims of conjunctival infections associated with Neisseria gonorrhoeae are infants or newborns delivered from infected mothers. The primary reservoir of Neisseria gonorrhoeae infections are asymptomatic carriers, or those men and women who have gonorrhea but are not showing any symptoms. The incubation period of gonorrhea ranges from 2 to 7 days. And the classic sign and symptom includes purulent discharge, meaning to say the presence of draining pus among men and women, especially pus that drains in the urethra and endocervix of men and women. And because there is pus as a result from the infection of gonorrhea to the urogenital tracts of men and women, then there would also be an accompanying dysuria or painful or difficulty in urination. For asymptomatic carriers, take note of this. The primary causative agent is the AHU strain of Neisseria gonorrhoeae. I'll repeat, for asymptomatic carriers, the primary causative agent is the AHU strain of Neisseria gonorrhoeae. So what is an AHU strain? AHU strains of N. gonorrhoeae require additional amino acid such as arginine, hypoxanthine, and uracil. They are oxotypes of Neisseria gonorrhoeae, and as I said earlier, they have been isolated as causative agents of asymptomatic infections in women and in men, and they are also isolated as causative agents of disseminated Neisseria gonorrhea infections. In women, the common site of infection occurs in the endocervix, which is characterized by the presence of cervical discharge, dysuria, and lower abdominal pain. However, take note that 50% of women are asymptomatic, and as I said earlier, this is caused by the AHU oxotype of Neisseria gonorrhoeae. Gonorrhea in women could complicate to pelvic inflammatory disease, or basically the inflammation of the pelvis as a result of the infection caused by Neisseria gonorrhoeae. And it affects one or more of the upper reproductive organs, including the uterus, the fallopian tubes, and the ovaries. And once there is PIV, sterility could occur, to which women having PIV would not be able to bear an offspring. And if they become pregnant, it would lead to ectopic pregnancy, wherein fertilized egg implants and grows outside the main cavity of the uterus. So basically, when a woman becomes pregnant, naturally, the fetus implants and grows in the uterus. But during ectopic pregnancy, it would be implanted in an area other than the uterus. Usually, it occurs in the fallopian tube. Perihepatitis is also another chronic manifestation of pelvic inflammatory disease. This is also known as the Fitzhugh-Curtis syndrome. This is an inflammation of the serous or peritoneal coating of the liver, as you can see in this picture, but without the involvement of the liver tissue. For disseminated gonococcal infection, or DGI, this results when Neisseria gonorrhoeae from the actual infection sites, such as the urethra of men or endocervix of women, disseminate into the bloodstream, causing septicemia. And from the blood, they further disseminate to distant sites of the body. And as I said earlier, the disseminated form of gonococcal infection is mostly associated with infections caused by the AHU oxotype of Neisseria gonorrhoeae. Clinical manifestations of disseminated gonococcal infection includes septic arthritis or a painful infection in the joint caused by the presence of Neisseria gonorrhoeae. And because there is septic arthritis, there would be joint pain, also known as polyarthralgia. The tendons are also affected, particularly the protective sheath surrounding the tendons. When Neisseria gonorrhoeae, particularly the AHU strain, disseminates, they would infect the synovial membrane that surrounds the tendon, and this would result to pyogenic flexor tenosynovitis. And this is its clinical manifestation. Pyogenic because it also accompanies PAS, or it is also accompanied with PAS. And the endotoxin component of Neisseria gonorrhoeae also leads to the formation of petechial or pustular skin lesions with fluid-filled pus. And the inner lining of the heart's chambers and valves also become affected during disseminated gonococcal infection. And this is what we call endocarditis. And when Neisseria gonorrhoeae is able to breach the blood-brain barrier, then they would also be able to cause infections of the fluid and membranes surrounding the brain and the spinal cord. Ophthalmia Unitorum is a conjunctival gonococcal infection that occurs among newborns delivered from infected mothers. And this could lead to blindness. So they are innocent victims of the situation. So take note that previously, Ophthalmia Unitorum is treated with Creed's prophylaxis using 1% silver nitrate. However, because it's a heavy metal and it's toxic, it is no longer used. And today, newborns are given erythromycin drops or antimicrobial eye drops as prophylaxis for ophthalmia neonatorum. Now let's talk about the laboratory methods for the diagnosis of Neisseria gonorrhoeae. So the primary sources of specimens for the diagnosis of Neisseria gonorrhoeae in the laboratory are genital sources, specifically purulent discharge from the urethra in men and endocervix in women, because these are the primary sites of infection. However, they can also be isolated from purulent discharges obtained from the rectum and pharynx for those men and women who practice unsafe oral anal sex. And joint fluids can also be collected for those patients with disseminated gonococcal infection. In men, when no apparent discharge or if discharges are not visible, the swab is inserted up to 2 centimeters in the anterior urethra and slowly rotated to collect the discharge. So I bet this is really painful. And take note that in specimen collection for Neisseria gonorrhoeae using swabs, 2 swabs must be collected. One swab is dedicated for collecting specimens to be subjected for direct microscopy, and another swab is dedicated for collecting specimens for inoculation on primary culture media. Take note, ladies and gentlemen, that it is a big no-no to utilize the same swab used for direct microscopy in inoculating primary culture media for the isolation of Neisseria species. For those with rectal gonorrhea, swabs for rectal culture should be inserted 4 to 5 centimeters into the anal canal. And take note that calcium alginate swabs and the ordinary cotton swabs that we use in the laboratory are not suitable for collecting specimens for Neisseria gonorrhoeae because they contain toxic substances that inhibit Neisseria gonorrhoeae. and therefore Dacron swabs are preferred. And as I said earlier, because of their extreme susceptibility to drying, because they require increased moisture and temperature changes, because they are susceptible to cold, specimens that are submitted for the isolation and speciation of Neisseria gonorrhoeae must be plated immediately to primary selective media. In this case, we are going to use enriched chocolate agar plate with inhibitors and added nutrients to support the growth of fastidious Neisseria gonorrhoeae. Blood specimens can also be collected to diagnose disseminated gonococcal infection. However, take note that Neisseria gonorrhoeae is inhibited by the presence of sodium polyanethyl sulfonate that is present as an anticoagulant in blood culture systems. And therefore, for this purpose, we can add gelatin to neutralize the harmful effects of SPS towards Neisseria gonorrhoeae. There are also commercial transport systems available that can be used to inoculate specimens collected from patients' bedside. We have here the Jembeck plates, also known as the James E. Martin Biological and Environmental Chamber, Transgrow, and the Gonopack. So I don't have a picture of the Gonopack. So these commercial transport systems contain selective media and can be transported with a CO2 atmosphere to provide optimal conditions until the specimen reaches the laboratory. So in the patient's bedside, if you collected a swabbed specimen, either from the endocervix of women or urethra of men, the swab containing the specimen should be rolled in a Z pattern on this medium. Rolling the swab through a Z pattern ensures adequate space for Neisseria gonorrhoeae to grow. And once these commercial transport systems are received in the laboratory, the media can be cross-streaked using a loop to facilitate the growth of isolated colonies. Once clinical specimens are received in the laboratory, direct microscopy should be done, especially from smears prepared from urogenital specimens, particularly endocervical and urethral specimens collected from women and men, respectively. However, take note that not all specimens received are subjected to direct microscopy, such as those collected from nasopharyngeal specimens, because as emphasized earlier, these anatomical sites may contain commensal bacteria, including commensal Neisseria, that may be mistakenly identified as pathogenic Neisseria. And therefore, when these specimens are received, they are inoculated immediately using an enriched chocolate agar plate that contain inhibitors. However, for urogenital specimens, once received, they must be subjected to direct microscopy. And take note, these are the significant findings that must be observed for clinical specimens collected from patients suspected of having Neisseria gonorrhoeae infection. There should be presence of gram-negative intracellular diplococci. When we say intracellular diplococci, these are gram-negative diplococci that are found within polymorphonuclear leukocytes. And their intracellular presence indicate that they are virulent because they possess pili that enable them to bind and enter leukocytes. However, we should also take note of the presence of extracellular gram-negative Diplococci, as they also indicate the presence of virulent forms or those Neisseria gonorrhoeae that already have lost their pili. So take note, these are the significant findings that would help us presumptively identify the presence of Neisseria gonorrhoeae. However, take note that since this is presumptive, it is not conclusive. Meaning to say that if we are able to observe the presence of gram-negative intracellular diplococci, it may suggest the presence of Neisseria gonorrhoeae, but it may also suggest the presence of Neisseria species and other bacteria sharing the same morphology and arrangement, and therefore it's only presumptive, and we need to proceed to culture so that we will be able to perform other tests that will help us definitely identify, or that will help us definitively identify the presence of Neisseria gonorrhoeae from these specimens. In culturing specimens for the isolation of Neisseria gonorrhoeae, take note, as emphasized repeatedly earlier, that Neisseria gonorrhoeae does not grow well on sheep blood agar because it lacks the nutrient requirements for fastidious Neisseria gonorrhoeae to grow, and therefore the medium of choice would be the enriched chocolate agar plate. So when we say enriched chocolate agar plate, although chocolate agar plates by themselves are already enriched because they have been added with blood that is lysed when heated at high temperatures to liberate X factor and other nutrients present in the red blood cells, they are further enriched with antibiotics and other supplemental nutrients to support the growth of Neisseria gonorrhoeae. And inoculated enriched chocolate agar plate are incubated at 35 degrees Celsius in a 3 to 5% CO2 because the organism is capnophilic. And because they require increased humidity, we should be able to provide sufficient humidity in the medium. And take note, if you are using a candle jar, the moisture evaporating from the media in a closed jar could suffice the needed humidity. If you are using a CO2 incubator, then it has already a built-in humidifier. But you can also use a pan placed with water and placed at the bottom of the incubator, as the vapor produced by the water present in the pan during incubation would also provide sufficient humidity. And take note that growth for Neisseria gonorrhoeae on enriched chocolate agar plates must be observed for 7 days. Meaning to say that if you were not able to observe growth in 24 to 48 hours, you should re-incubate it until 7 days before reporting culture as negative. Because take note, there are oxytypes of Neisseria that are slow growers, such as the AHU strain, that require more days of incubation before they are able to produce colonies. So as I said earlier, an enriched chocolate agar plate is needed to support the growth of Neisseria gonorrhoeae. And these are examples of enriched chocolate agar plates that can be used in the primary isolation of Neisseria gonorrhoeae from clinical specimens. So take note that Thayer-Martin and modified Thayer-Martin media are similar in many ways. Basically, they are similar in terms of nutrient contents that support fastidious Neisseria gonorrhoeae. Both Thayer-Martin and modified Thayer-Martin media contain hemoglobin as a source of X factor. It is also enriched with V factor, vitamins, amino acids, coenzymes, and dextrose. And in terms of inhibitors, both media contain vancomycin that inhibits gram-positive bacteria, colistin that inhibits other gram-negative bacteria except itself, because Neisseria gonorrhoeae is also gram-negative, and nystatin that inhibits fungi. So what makes modified Thayer-Martin different from Thayer-Martin is the addition of trimethoprim lactate that inhibits swarming of proteus. So this image here shows swarming colonies of Proteus species. And when this phenomenon occurs, the swarming growth could hide colonies of Neisseria gonorrhea or even Neisseria meningitidis, so to speak. And in order for us to inhibit swarming of Proteus so that colonies of Neisseria gonorrhea and Neisseria meningitidis could be well visualized, then we can use modified Thayer-Martin medium as it contains trimethoprim lactate that inhibits swarming of Proteus. We also have here Martin Lewis and New York City agar. So both of these media contain virtually the same inhibitors, but Martin Lewis contains anisomycin as inhibitor for fungi, and New York City agar contains amphotericin B as inhibitor for fungi. And in terms of the nutrients present, Martin Lewis medium contains bovine hemoglobin that provides X factor, nitrogenous nutrients in the form of casein and meat-peptone, and phosphate buffer to maintain pH. And take note, it contains cornstarch that neutralizes toxic fatty acids present in the medium. It is also enriched with V factor, vitamins, amino acids, coenzymes, dextrose, ferric ions, and other growth factors necessary for improvement and optimal growth of pathogenic Neisseria species. Then we have for New York City agar, it contains peptone, cornstarch that neutralizes toxic metabolites produced by Neisseria, horse plasma, horse hemoglobin, dextrose, and yeast autolysate. And take note, it also has sodium chloride that provides essential electrolytes, maintaining osmotic equilibrium, thereby maintaining the integrity of cells. And take note that this media we have here, Thayer-Martin, modified Thayer-Martin, Martin Lewis, New York City agar, all of these media are enriched chocolate agar plates that support primarily the growth of Neisseria gonorrhoeae from clinical specimens. However, it can also be used to isolate Neisseria meningitidis if the specimens obtained are coming from anatomical sites that are normally populated with microbiota. Once culture and incubation is done, then we can observe for the presence of growth. Typically, a 24-hour or 48 hours of incubation of culture for the isolation of Neisseria gonorrhoeae would reveal the presence of small gray to tan, translucent, and raised colonies. So these are the colonies that can be obtained from a fresh culture of Neisseria gonorrhoeae using enriched chocolate agar plates. Specifically, there are 5 types of colonies that can be produced by Neisseria gonorrhoeae on culture media. We have T1 and T2 and T3 to T5. So T1 and T2 colonies, these are the fresh colonies produced within 24 to 48 hours of incubation and characterized by being small and raised and appear bright in reflected light. And these colonies are pileated, meaning to say the organisms growing here contain pili and are considered virulent colonies. However, subsequent incubation of culture for Neisseria gonorrhoeae would result to the progression of T1 and T2 colonies to become T3 to T5 colonies, which are already non-pileated, meaning to say that the bacteria growing already have lost their pili due to subsequent incubation and have now become virulent. And these colonies are characterized as larger, flatter colonies. AHU colonies, when present, produce smaller colonies and grow slowly, and this is for the reason why culture for Neisseria gonorrhoeae must be incubated further until 7th day of incubation before reporting a negative culture. And take note, this is very important, that when you do work up, a fresh culture must be used since older cultures can no longer contain viable Neisseria gonorrhoeae since they have their ability to produce their own autolytic enzymes. And when Neisseria gonorrhoeae produces autolytic enzymes, they can lyse themselves. And this could result after subsequent incubation. So take note, fresh culture or colonies obtained from fresh culture is recommended for workup because, as I emphasized earlier, older cultures can result to the production of their own autolytic enzymes that can lyse their own cells. And when you use old cultures for workup, then you might not be able to obtain reliable results in the biochemical test for Neisseria gonorrhoeae identification. So once colonies are obtained, you are going to perform indirect microscopy. When we say indirect microscopy, you are examining the colonies grown on enriched chocolate agar plates used to grow Neisseria gonorrhoeae from clinical specimens. And in doing such, you must be able to observe the presence of gram-negative diplococci that are bean or kidney shaped. So these are the typical appearances of Neisseria gonorrhoeae on indirect microscopy. So notice the presence of gram-negative diplococci that are kidney or bean shaped. However, take note that this is just presumptive, meaning to say the appearance of gram-negative diplococci that are bean or kidney shaped may mean the presence of Neisseria gonorrhoeae, but we cannot conclude because there are other Neisseria species that may also have the same morphology and arrangement. And take note, Kingella and Acinetobacter may occasionally grow on the enriched chocolate agar plates used to isolate Neisseria gonorrhoeae from clinical specimens, but we should not be confused by this, as these bacteria are rod-shaped, while Neisseria gonorrhoeae organisms are diplococci that are bean or kidney-shaped in appearance. Take note that these findings in indirect microscopy, namely gram-negative diplococci, bean or kidney-shaped, are the important findings that must be established before performing the presumptive biochemical test for the confirmation of the presence of Neisseria. And that presumptive biochemical test is the cytochrome oxidase test. I'll repeat, when shall you perform cytochrome oxidase test? Only if you are able to observe the presence of gram-negative diplococci that are kidney bean or coffee bean shaped in appearance. So the cytochrome oxidase test determines the presence of cytochrome oxidase enzyme in the cell membrane of Neisseria. And the cytochrome oxidase enzyme of the organism reacts with oxidase reagent. And in the presence of atmospheric oxygen, it would result to the formation of an indophenol compound. So there are two reagents that can be used, either of these two. We have 1% dimethyl, paraphenylene diamine dihydrochloride, or 1% tetramethyl, paraphenylene diamine dihydrochloride. So in performing the test, we can use filter paper discs that are smeared with colonies containing gram-negative diplococci that are kidney bean or coffee bean in shape. And these colonies containing those morphological features will be added with any of these oxidase reagents. And a positive reaction is indicated by the appearance of a purple color within 10 to 30 seconds. And if you have this reaction, then this is reported as positive for oxidase test, and it confirms the presence of Neisseria species. Take note that we are still in the genus level. since all members of the genus Neisseria are oxidase positive. But it is good to know that we are now closing in to the isolation of Neisseria species and are eliminating other organisms that are also gram-negative diplococci. So since this is just presumptive and only confirming the presence of Neisseria, we are not going to stop here, but instead we are going to perform the definitive test to speciate pathogenic Neisseria. And that test is what we call carbohydrate utilization test using CTA medium. However, before we go to that, take note before I'll forget that apart from performing oxidase test on a filter paper, we can also perform oxidase test alternatively by dropping the oxidase reagent on the colony that contain gram-negative diplococci that is kidney bean or coffee bean shaped. And if the colonies turn deep purple to black, then it's positive for Neisseria species. So going back, as I said earlier, since oxidase test is presumptive only for the presence of Neisseria and cannot help us identify which pathogenic species is present, then we can proceed to the definitive tool for identification, which is carbohydrate utilization test using the CTA medium or the cysteine trypticase agar. So these media here are what we call the CTA media or the cysteine trypticase agar. And these are fermentation media to determine the action of Neisseria species towards any of these carbohydrates present, whether they are able to ferment glucose, maltose, lactose, or sucrose. So as fermentation media, these tubes here contain the indicator phenol red that changes its color from red to yellow in the presence of an acidic byproduct. And each media here contain 1% of individual glucose. So one tube contains 1% glucose. Another tube contains 1% maltose. Another tube contains 1% lactose. And another tube contains sucrose. And as a review, when carbohydrates are utilized, mixed acids will be produced, dropping the pH, resulting to a change of the color of the phenol red indicator from red to yellow. For Neisseria gonorrhoeae, take note that they are only able to ferment glucose. And if you have a positive glucose fermentation test using the CTA medium indicated by the presence of a yellow color in the tube that contains glucose, then this is already definitive for the presence of Neisseria gonorrhoeae. So in this way, we can now speciate and we can now perform antimicrobial susceptibility testing to determine which antibiotics are effective or not effective for the treatment of Neisseria gonorrhoeae. Carbohydrate utilization test can also be done using other methods. We have here chromogenic substrate method. So this is a test strip that contains wells, and each well contains carbohydrate substrates. And if Neisseria species are inoculated, the bacterial enzymes produced by these organisms would hydrolyze the carbohydrate substrate present. And in this case, since we are talking about Neisseria gonorrhoeae, it only produces a chromogenic reaction in the well that contains glucose because Neisseria gonorrhoeae only ferments glucose. So this is useful for the identification of Neisseria gonorrhoeae with aberrant carbohydrate utilization reaction in CTA. We also have this multi-test method. So this is a method that follows the conventional chromogenic enzyme principle, but this is a combination of other characteristics. So apart from we are able to determine carbohydrate utilization, we can also be able to identify whether the organism possesses DNAs that hydrolyze DNA. and beta-lactamase that hydrolyzed beta-lactam ring of beta-lactam drugs. And take note that aside from this can be used to identify Neisseria gonorrhoeae from clinical specimens, we can also use this multi-test method to identify other genera of bacteria, such as Haemophilus species and Moraxella species. All right, so as discussed earlier, once we are able to isolate Neisseria gonorrhoeae, definitively using carbohydrate utilization test, then we can proceed to the performance of antimicrobial susceptibility testing to determine whether or not the antibiotics tested would be effective in treating Neisseria gonorrhoeae infection. However, apart from performing antimicrobial susceptibility test, we can also determine the ability of Neisseria gonorrhoeae to produce the beta-lactamase enzyme. So to understand this test, we should learn first what is beta-lactam and what is beta-lactam ring. When we say beta-lactam drugs, these are antibiotics that contain a beta-lactam ring in their structure. And these antibiotics containing beta-lactam rings in their structures include penicillins, cephalosporins, penims, cephins, carbapenims, and monobactams. So the beta-lactam ring, as I said earlier, which is the structure present in these beta-lactam drugs, contain this beta-lactam ring. So this is the beta-lactam ring. And you'd know it's the beta-lactam ring because it has a nitrogen atom in it. And what does this beta-lactam ring do? It binds to the penicillin binding proteins of the peptidoglycan layer of both gram-positive and gram-negative bacteria. And the binding of beta-lactam ring to the penicillin binding proteins would result in the inhibition of cell wall synthesis. And when this occurs, bacterial lysis and death happen. So I have here a visualization. So this picture here shows the beta-lactam ring of beta-lactam drugs. And as I said earlier, you'd know it's the beta-lactam ring because of the nitrogen atom. And this is the key to the antibiotic property of beta-lactam drugs. So what does this beta-lactam ring do? So let's focus here. Naturally, the PBPs, also known as transpeptidases, helps in the cross-linking and polymerization of peptidoglycan strands to complete peptidoglycan synthesis. If beta-lactam drugs are used to treat the infection caused by bacteria, the beta-lactam drug, particularly its beta-lactam ring, would bind to the PBPs. And this binding inhibits the function of PBPs, which is to polymerize and cross-link peptidoglycan layers. And when there is failure of the PBPs to cross-link and polymerize peptidoglycan layers, then cell wall synthesis would be halted or inhibited, and this results to bacterial lysis and death. Now, where does beta-lactamase enzyme come into play? Take note that there are bacteria that possess beta-lactamase, also known as penicillinase enzyme. Both gram-positive and gram-negative bacteria may have the ability to produce this enzyme. And if they have this enzyme, which is beta-lactamase or penicillinase, this enzyme hydrolyzes the beta-lactam ring, making beta-lactam drugs ineffective. And those bacteria that produce beta-lactamase are said to be resistant to penicillins and other beta-lactam drugs. So examples of bacteria that produce beta-lactamases or penicillinases include, but not limited to, Neisseria, Escherichia coli, and Staphylococci. So I have here a visualization. So naturally, as I said earlier, the penicillin binding proteins act in the polymerization and cross-linking of peptidoglycan layers, resulting to a successful synthesis of cell walls. However, naturally, the beta-lactam ring of beta-lactam drugs should inhibit PBPs from doing such. But for those bacteria that possess beta-lactamase enzyme, they will bind to the beta-lactam ring, hydrolyzing it, and therefore there would be no beta-lactam rings that would inhibit the penicillin binding proteins, and therefore cell wall synthesis could occur. And as I said earlier, those bacteria that produce beta-lactamase are said to be resistant to penicillin and other beta-lactam drugs. So take note that if this scenario happens, beta-lactam drugs such as penicillins and cephalosporins would no longer be effective in treating infections caused by beta-lactamase producing bacteria. So in the laboratory, we can screen bacterial isolates for the presence of beta-lactamase enzyme. And we do this to determine whether or not penicillins and cephalosporins, which are beta-lactam drugs, are still capable of inhibiting these types of bacteria. So for the principle of beta-lactamase test, the test determines the ability of the bacterial isolate to produce the beta-lactamase enzyme that hydrolyzes beta-lactam rings, making them resistant to penicillins and other beta-lactam drugs. So the method used is the Cephinase test, and this test detects beta-lactamase production from colonies of Staphylococcus species, Haemophilus influenzae, and in this case, Neisseria gonorrhoeae, and anaerobic bacteria as well. And take note that isolates containing these organisms should be routinely tested for beta-lactamase activity to determine whether or not penicillins and cephalosporins are still capable of inhibiting them. The reagent is Cephinase disc, and these are the Cephinase discs. These discs are impregnated with a chromogenic cephalosporin. Chromogenic cephalosporin contains the beta-lactam ring, which serves as the substrate of the test. And if these isolates here contain beta-lactamase, then they would be able to hydrolyze the beta-lactam ring of the chromogenic cephalosporin, resulting to a color change. So for the procedure, the Cephinase discs are moistened with a 1 drop of purified water. And using a sterile loop or needle, we are going to obtain a colony from the speciated isolate and smeared on these discs. And if the colonies that have been inoculated or smeared in these discs are able to produce the beta-lactamase enzyme, then they would be able to hydrolyze the beta-lactam ring of the impregnated chromogenic cephalosporin, and this would result to a change of the color of the disc from yellow to red. And if this is the reaction, then the isolate is positive for beta-lactamase. And when it is positive, take note that beta-lactam drugs should no longer be prescribed in treating Neisseria gonorrhoeae bacteria and other bacteria that tested positive for beta-lactamase. So in terms of antimicrobial resistance, take note that previously Neisseria gonorrhoeae is treated with penicillin. It is actually the standard treatment used to treat Neisseria gonorrhoeae. However, the emergence of penicillinase producing Neisseria gonorrhoeae, or those Neisseria gonorrhoeae that is able to produce beta-lactamase, they have now become resistant to penicillin, and take note that penicillins are no longer used as a routine treatment method for Neisseria gonorrhoeae. The ability of Neisseria gonorrhoeae to resist penicillin by means of its ability to produce penicillinase or beta-lactamase could be plasmid mediated, and it is transferred by one organism to another through the use of the conjugation pili. Apart from resistance to penicillin, there is also what we call plasmid-mediated and chromosomally-mediated resistance of Neisseria gonorrhoeae to tetracyclines, pectinomycin, and fluoroquinolones. Currently, for those isolates that are not positive for beta-lactamase, cephalosporins remain the current recommended treatment. And on top of that, CDC recommends the use of ceftriaxone, cefexime, ciprofloxacin, ofloxacin, and levofloxacin as standard treatment methods for uncomplicated gonorrhea. Here ends part one of our discussion on the family Neisseria. Later on, I will discuss Neisseria meningitidis and the rest of those organisms that are related to the family Neisseria.